This week, I believe there'll be no seminar as it is the Research Computing Symposium next Thursday, so you're all encouraged to go to that, and then we'll meet up again the following Thursday. There is a sign-in sheet. We'll pass that around. If you could please sign in, it helps for the pizza and just so that we know who's attending. And with that, I'll go ahead and turn it over to our speaker. Today we have Ridvan Eksi, who's going to talk to us about IsoPred.
Thanks, Marcy, for introduction, and thanks for giving me this opportunity to present here in this seminar. So today I'll talk about this paper that is published a while back. I believe it's a useful tool for many of you in your own studies. So it's called IsoPred. It's systematically differentiating functions for alternatively spliced isoform through integrating RNA-seq data. So this is the outline of my talk. So I'll first give some introduction and talk about why is it important to do functional prediction studies, and particularly why it is important to do for alternatively spliced isoforms. And I'll talk about the methods and the data, and I'll introduce multiple instance learning that we use. Then I'll show the results and the validations that we did, and summary, and also last I will show the web server that we released the results. So the accurate annotation of protein function is a key topic in biology for understanding of diseases and molecular processes, and experimental approaches for functional annotation such as like knockout or knockdown experiments are not feasible for genome-wide studies. So that's why there is this whole bunch of computational function prediction studies. But most of these studies are done on gene level. So for a specific function, usually defined by these gene ontology terms, genes that are already known to have this function are called as positives and the rest as negatives. And you have a machine learner, you construct a model by using some kind of genomic features, and you do your functional prediction. But the major limitation of this kind of function prediction studies is that the gene is considered a single element. But we know that it can be spliced into different transcripts, resulting in proteins maybe carrying different functions. So there are multiple ways of multiple mechanisms of alternative splicing, and each can produce a different different mRNA sequence and different protein sequence from a single gene. So if you look at the numbers in current annotation packages, so if this is from GenCode, there are around 57,000 genes and close to 200,000 transcripts from those genes. If you look at the protein coding genes, around 20,000 genes and 80,000 transcripts. So that means on average there are 4 transcripts per gene. So when you do your functional studies on gene level, you're missing out this another level of resolution. So it's estimated that 95% of multi-exon genes undergo alternative splicing. So these numbers are not the complete numbers. So there are a lot more transcripts to be discovered. And the splicing plays a role in post-transcriptional gene regulation, or it can alter the protein sequences and domains, it can modulate regulatory elements, so it can have functional consequences. And so here I have 4 examples of transcripts from the same gene with different functions. So this TRPM3 gene, which encodes a cation selective channel in human, and it has 2 variants targeting different ions. And the variants of BCLX gene, one of them is anti-apoptotic and the other one is pro-apoptotic, so opposite functions from the same gene. And same for Cas3L and Cas3S variants, also same for OSR2 gene. So these two transcripts from same gene can have different or even opposite functions. So attempts to capture this kind of functional differences of transcripts are limited to low throughput experiments and also some computational predictions through domains or binding region or binding sites. But one challenge facing the experimental studies are that generally isoform specific antibodies are not available. So you cannot do Western blot or ChIP-J, so they don't routinely provide resolution at the isoform level. Also, isoform specific oligonucleotides are difficult to design for qPCR or RNA interference because you have very small changes between the isoforms. So also for computational studies that are doing domains are limited in a sense that only 34% of the isoform pairs of the same gene differs in terms of domains. So the remaining are exactly they have same set of domains, so you cannot really differentiate. So in this study, we are trying to overcome some of these difficulties. So the first limitations that we face are, the two limitations that we faced are first, the functional genomic data are generally available for gene level, at the gene level, such as microarray dataset or most of the protein-protein interaction datasets. And the other limitation is that functional annotations are centered on genes. So in gene ontology or in KEGG, you see the annotations based on genes. So we'll try to overcome these limitations with our approach. So we use RNA-seq data because it can give transcript resolution data. So we use publicly available mouse RNA-seq experiments from NCBI's SRI database. So as of November 2012, there were around 1900 experiments. It's increasing day by day, but the time we started with this many experiments, and this includes a lot of different tissues, different experimental conditions, and also includes some part of the ENCODE RNA-seq data. And we processed them using Tuxedo-seq. After trying out different tools, we decided to use Top Hat Cufflink's pipeline to get the abundance estimates for transcripts. And we used NCBI's annotation package, which has 20,000 genes and 30,000 transcripts. So we did some quality control for the RNA-seq data and ended up using 365 experiments in this study. So this was the first. So we now have functional genomic data on transcript level. So the second limitation is the gold standards. So for that, to overcome this, we used multiple instance learning. So this is a kind of a semi-supervised learning approach that has been introduced in 90s, I guess. So in this one, so instead of having genes as a single point in your classification, we consider genes as a bag of transcripts. So here I have, this is a regular, let's say a classifier. You have a separating hyperplane, and you're trying to maximize the distance between these points and this separating hyperplane. So here each dot represents a gene for previous functional studies. In our study, we have individual transcripts that are labeled by their genes. So instead of having label for each point, we have a label for each bag, each collection of instances. So the idea is to extract the common patterns of isoforms across positive genes, then use these patterns to find which subset of these positive genes, positive transcripts are actually positive, and then use this to do the classification. So there are two assumptions for multiple instance learning. So if a gene is positive or a bag is positive, at least one of its isoform will be positive. So if you know that a bag is positive here, then at least one of its isoforms should be positive. And also if a gene is negative, then all of its isoforms will be negative. So there is no ambiguity about the negatives. Only is which subset of isoforms of a positive gene will be positive. That is the ambiguity we're trying to solve. Just a toy example. You have two positive genes. Positive means this gene is annotated to a specific function that we studied. So let's say gene A and gene B are both positive, and this one has three transcripts, this one has 2 transcripts. So there are 14 possible assignments of labels to these genes. So multiple instance learning tries to find the optimum assignment in this combinatorial space. So this is the kind of overall pipeline. So we have an initial assignment of isoform function pairs. It's just in the beginning we assign every transcript of a positive gene as positive. So that's our initial assignment. So then we build our classifier based on that and reclassify our input set. And then we have a new set of labels. And then we refine this new set to be consistent with these assumptions. And then we reclassify. So this iterates until there is no label change. And then this will converge at one point, and we will have the optimum labeling of transcripts after this step. And then we can go ahead and build our final classifier to get the probability that isoform is annotated to a function or probability that a gene is annotated to this function. And isoform abundance estimates are our input features. So there are different formulations of multiple instance learning. The first approach is to, let me show you in this figure. So this is a positive gene with multiple transcripts. So one of the approaches is just choosing one of the transcripts as witness and discard the others. So instead of having this many transcripts, we will only have one positive that is representative of that gene. And the other approach is to have a threshold and have a group of transcripts as positive and the rest as negative. So you can choose different threshold resulting in different ratios of positives to negatives. So we ended up choosing like a mild approach. So this is the most strict version. You only have one isoform left, and this is kind of less strict version. You have a lot of positive transcripts. So we ended up choosing somewhere middle approach because it was giving best performance based on cross-validation. So after that, we run our predictions, and we have several lines of validations, including computational validations and some specific examples of genes. So for all of these validations, we tested around 1,800 biological terms with 20 to 300 genes annotated to them. So for the terms that have less than 20, they're too specific to do any prediction, or for the ones that have more than 300 genes annotated to them are too broad to be included in this study. So, and also we grouped Go terms in based on their sizes, based on their number of genes annotated to them, and we reported AUC, AUPRC, and other metrics to show the performance. So the first one is cross-validation. So in this one, we partitioned the genes into 2 groups. One test set held out and training set, we build the model based on training and test the holdout set. These are the AUC values for 5 different groups of Go terms. So the AUC is around 0.67, 68, which is comparable to previous gene function prediction studies that used multiple data sources. So we achieved this AUC just by using expression data and by using this multiple instance learning. And there is also a trend that the bigger Go terms are easier to predict than the smaller terms. We can see the same trend for AUPRC too. So that was the cross-validation. But in this figure, the performance is the mix between single isoform genes and multi-isoform genes. So since our framework is kind of making a difference for multi-isoform genes, we wanted to see if we have like a performance gain between multi-isoform genes and single isoform genes by using these. Because for single isoform genes, there is no ambiguity. It's just same as before, just regular supervised classification. So we expect to see a better performance for multi-isoform genes because, as I said, the list of transcripts are not complete. So most of the single isoform genes are not really single isoform genes, but they are the ones that haven't been, the other transcripts haven't been identified yet. So that will cause some performance drop for these single isoform genes. If we look at the performance, AUC and AUPRC, for all Go term groups, there is a consistent performance gain by multi-isoform genes compared to single isoform genes. That was another line of validation for us, and that showed us that multiple instance learning does bring some discriminative power. And we also tested the algorithm with respect to two cases. So first, we compared the estimates of performance of algorithm on the genes that are highly expressed versus on the genes that are lowly expressed, because we know that this RNA-seq experiments, the abundance levels estimated from RNA-seq experiments will be less reliable for low expressed genes. So we divided the genes into three groups based on their expression, like high expression group, medium expression group, and low expression group, and show that, well, although the high expression group performs better, there is no big difference between them. So the algorithm is robust based on their genes expression value. And the second one, the homolog gene pairs. So when we're dividing our gene set into training and testing, we might have some examples of one gene in training set and its homolog gene in test set. So that can cause some overfitting or some information leak, and that may increase the AUC values. So we tested in a way that none of the homolog pairs will be both in training and testing set. So if there are homolog pairs, they'll both be in training or they will both be in test set. So again, there is a little bit difference, but not much. So it is also robust to this information leak. So up to here, I showed you some computational evaluations. So here, I'll show some proteomics data that are that is also another way of evaluating the results. So our hypothesis is that, well, we see that most of the cases, one of the isoforms is functional. So when we look at the predictions, most of the cases, one isoform is doing the function, or have high probability of having the function for most of the Go terms. So we wanted to check.
Are you excluding all long non-coding transcripts?
No. Well, but in this set that we use, in the NCBI set, there aren't a lot of them. But we don't include we don't exclude any non-coding.
They don't get expressed at the protein
Yeah.
level.
Well, but as I said, there aren't a lot of examples of long non-coding transcripts in NCBI. Also, the ones that exist get filtered out based on the expression, because they're not expressed in most of the experiments that we process. They're not actually they are not included in the analysis, most of them. So the idea is that if a transcript is functional, then at the protein level we should be able to see it in the tissue. So we first identified the isoform which is strongly expressed, and also we choose among those genes, we chose the ones that have a known function, and also their isoform are predicted with this drastically different confidence level to carry out this known function. So all the expressed isoforms are predicted with the highest value in at least one of the known functions. So there is a consistency between the predicted functional isoform and the expressed isoform. So here we have 6 genes and several Go terms. In almost in all of them, the predicted isoform by our algorithm is the one that is found in the proteomic sample. So that was another line of validation for us. So now I'll talk about these 2 specific genes that we choose. So it's not just identifying the functional isoform, but the algorithm can also predict different functions for each one of the isoforms. So we focus on analyzing these 2, and so this CDKN2A gene is a gene where alternative splicing results in different reading frames. So this is an example where we have 2 very different proteins that are coming out of this gene. So this is the first isoform, second isoform. They share an exon, but since there is a frame change, the proteins that are encoded by these transcripts are really different. So these are the structure prediction by iTesser. So different domains and different shapes, and we expect to have different functions for them. And if you look at the predictions, the first isoform is predicted to be functional for this function, and second one is predicted to be functional for the second function, for this term. And surprisingly, both of them are predicted to be functional for this term.
What's the number mean?
Which number? Oh, so that is the
Yeah, thanks. I didn't know.
fold change. So if we, let's say, if this term has 200 genes annotated to it, the baseline 200 over the number of genes, that is the baseline probability of a gene having this function. So this number is the fold change of the probability that we calculated. So it's 73 times more likely that this isoform will perform this function over baseline probability.
So does your prediction agree with iTesser's function? So with this term prediction, is it from iTesser results or from your pipeline?
No, this is from my pipeline. So this structures are from my pipeline.
I mean,
but
iTesser
we
also
don't.
iTesser also gives out like cofactor prediction on Go term. So does it agree with
No,
each other?
we didn't include anything from iTesser in the prediction step, in the functional prediction step. It's just a abundance estimates. So, so this is an example of a gene where 2 transcripts are drastically different. So it's expected to be, they are expected to be have different functions. But so yeah, these functions, all of the functions that we showed here are also mentioned in some literature. So this transcript is an inhibitor of CDK4 kinase, a member of this protein kinase family, supporting its role in this function. Yes, not here. That's second function. And the other isoform is a enhances p53 dependent transactivation and apoptosis, supporting its role in apoptotic nuclear changes. And although coding for dissimilar proteins, both share a common functionality in cell cycle G1 control. So our second example, ANXA6 gene, this one, two transcripts are very similar. The only difference is the 6 amino acids at these locations, and the rest of the proteins that are encoded by this transcript are same. So the positions of threonine 535 and serine 537 in one of the transcript compared to other make this one more likely to go phosphorylation because of the structure change in this part. So the fold changes on Go terms related to phosphorylation by our function prediction algorithm supported this conclusion. So for example, for the Go terms peptidyl serine phosphorylation, this one has a higher probability of than the second one. So in summary, gene functions are ultimately delivered through their alternative splice transcripts that encode proteins with different functions, and it's highly beneficial that these functional studies is carried out at the isoform level. So, however, algorithmically, any supervised learning algorithm developed for this gene function prediction cannot be directly applied because we don't have gold standards. So we chose to use this multiple instance learning strategy to predict functions at the individual isoform, for the individual isoforms. So we, through several lines of validation, we show that it's producing promising results. And we released our...
Oh, can you go back? I just, you were going faster than I could read. Okay, thanks. I'm sorry.
No. So,
This, you should go through all of them. I mean, this is important stuff. Okay, so,
so yeah, this idea of applying this multiple instance learning to this problem is kind of is the novel idea in this study. So it has been used, multiple instance learning has been used in a lot of other different fields with success that are similar to this problem. And this isoform function prediction problem is like a perfect match for multiple instance learning framework because the assumptions holds true for this problem too. So we released our predictions in this website. I'll show some of the examples how to use this website. So you can do search with genes or with isoform names or with Go terms. For example, this is the one that I showed in the slides. So it gives a like an information about the transcripts, and it gives PFAM domains informations, and it gives the predictions for each Go term sorted by the maximum fold change. So here we see that this one is highly, well, this one is more likely to be functional for this term, or we can sort based on the second isoform too if we want. Or we can look at the Go term page where we see the more likely functional genes or isoforms for this go term. I want to show another example. This gene, the proteins are similar length, but if you look at the predictions, for some of the terms, both of them are predicted to be functional. For some, the second one is functional. For some, the first one is functional. So for most of these examples, we couldn't find any evidence from literature, or we don't have the means to test it out. But yeah, these are really interesting predictions that are showing very distinct predictions. So
Okay, so I don't know, maybe I'm just slow. I think a little bit more explanation about, this looks like a fantastic resource. That's, and the work is really wonderful. Where I was having trouble is just understanding, how you use this as a discovery tool. I mean, just at the beginning of the process, and, if you could take us through maybe one more example, it might become clear. Because this, there's lots of areas now in our area of psychiatric pharmacogenomics. I mean, alternative splicing is the name of the game in the brain. There's absolutely no question about it. And part of what we haven't been really looking at enough, frankly, is that we do this reanalysis and find structure mapping based on some of your work, Alan. And then we find results that are similar to what you find, Steve. That you have intergenic SNPs and intronic SNPs. We haven't spent much time on the intronic SNPs. We focused more on the intergenic side to follow your regulome concept. But there's, especially with bipolar disease, we're seeing a lot of new intronic SNPs that most likely are driving, different splicing isoforms. I mean, that's our working hypothesis right now. So, so I guess, as a bioinformatician that's got another tool in the toolbox, yours, if we have, some introns that we would like to look at, with some diseases in mind, tissue specific, kind of driven by Allen Brain Atlas and the NIH Epigenome Roadmap, how would we take that data into your tool and then get predictions of,
So here
So I'd like to try it.
So the predictions that we released here are only for the known transcript or annotated transcripts that exist in the NCBI database. But if you have a novel transcript that we discovered, let's say that discovered in the study, and if you have a kind of expression or abundance data for those new isoform, we can potentially run this isoform transcript through our pipeline to get the predictions for that too. But first we need to get this list of novel.
Right. So I guess part of my question is that, and I had it in my mind earlier. I guess part of my question is trying to, and forgive me, I came in late. You might have had this all. What was the knowledge base that was underlying all this? Because that's driving, the space of possibilities for, the use of this tool.
So the novel transcript discovery is putting another level of complexity. So to avoid that, we started with the known transcripts. Actually, we use the conservative set of known transcripts. So Ensemble has a lot more transcripts than NCBI, but we wanted to start with NCBI to be more conservative in the first study to see if it is really working. Then we can continue expanding our list of transcripts. So with that, I wanna acknowledge my advisor, Yongfang Guan, and the members of Guan Lab. Also, I want to thank to our collaborators, Gil Oman, Raji Menon, and Matthias Kretzler. And I'm happy to take any.
Well, and it looks like you've got some collaborators with SonLab too, with the iTaser.
Oh yeah, I want to. Yeah, I forgot to include. Yeah,
Right? I didn't want to forget you guys. Well, it's good collaboration there, you know? It shows what can happen up and down the hall. I mean, that's kind of cool. Any other questions? Brendan.
So if I'm understanding correctly, you have a list of Go terms that are associated with a gene. You sort of disambiguate which Go terms are associated with which isoform, right? So it's using that annotation beforehand, right? How do you? What element of the model is determining which Go term associates with it?
Okay,
This is
so
a major conceptual point.
It's a major point.
Yeah, I just want to understand.
so each prediction, well, each Go term is tested separately. So it's not like you're searching for which Go term is related to which isoform, but you're taking one Go term and you're doing the predictions for that term first. And
Sure.
you're doing this for every one of them separately.
Are you're using like domain information on
No,
each isoform?
this is
Okay.
just expression.
Oh, I see. Okay.
So the idea is if two isoforms have similar patterns, expression patterns, they're more likely to be co-functional. Okay.
Just one more question I had was on the machine learning. I mean, you know, the world isn't always black and white, so is there a way to put a fuzzy boundary on some of this and some probabilities instead of making the assignments just positive or negative?
We're making positive and negative for only gold standards. Actually, the negative set is not actually a negative set. So for the positives, they're really conservative. We include only the ones with strong evidence in positive set. But for negative, there are some genes in the negative set that are yet to be discovered. So we are actually downweighting the negatives because also it's very unbalanced. So you have, let's say, 300 positive genes and the rest are negative. So it's very imbalanced. So negatives are actually downweighted to have less impact on the classification. But for positive, we really treat them as positives.
And so the training set and gold standard are really, you know, there's an advantage and a disadvantage in it.
Yes.
The advantage is that, you know things, you want to find other things like it. The disadvantage would be that maybe, there's whole classes of things that don't even fit into the model that you had put in place. Okay, any more questions? Well, thank you. It was very nice.
For coming.
Ellen, how are you? I've been meaning to ask you how you doing.